Colorectal cancer is the third most prevalent cancer globally, and early detection is crucial for improving patient outcomes. A groundbreaking development in cancer detection is on the horizon, thanks to an international team of researchers who have engineered bacterial biosensors capable of identifying cancer-associated DNA mutations. This innovative technology could potentially revolutionize the early diagnosis of colorectal cancer, a leading cause of cancer-related mortality worldwide. In this article, we explore the remarkable advances made in bacterial biosensors for cancer detection and their potential impact on the field of medical diagnostics. Harnessing bacteria for cancer detection. The human gut is teeming with a diverse population of microbes that play essential roles in digestion, vitamin production, and immune system communication. Now, scientists are entrusting bacteria with a new mission: detecting cancer. This cutting-edge research, published in the journal Science, showcases the collaborative efforts of scientists from various disciplines, including molecular biologists Robert Cooper and Jeff Hasty from the University of California, San Diego, and bowel cancer researchers Josephine Wright and Susan Woods at the South Australian Health and Medical Research Institute, along with Daniel Worthley from the Colonoscopy Clinic. The primary objective of this research is to develop a bacterial biosensor capable of identifying a common colorectal cancer marker, a mutation in codon 12 of the KRAS gene. Colorectal cancer ranks among the most prevalent and lethal cancer types worldwide. By focusing on early detection through bacterial biosensors, the research team hopes to provide a means for more effective and timely diagnosis. A milestone in biosensor technology. While previous studies have explored the use of engineered bacteria to detect inflammation or bleeding in the gut, this research marks the first time that bacterial biosensors have been designed to identify a specific DNA sequence originating from host tissues. The researchers achieved this remarkable feat by utilizing the natural competence of Acinetobacter bailey, a bacterium with the ability to take up extracellular DNA and integrate it into its own genome. The experimental process involved genetically modifying both A. bailey and the tumor organoids that the bacteria were intended to detect. By engineering the tumor cells with a functional copy of the antibiotic resistance gene, KNR, flanked by KRAS homology arms, the researchers allowed the bacteria to integrate the KNR gene into their genomes. This enabled the bacteria to grow on antibiotic-containing plates, providing a clear signal of the tumor's presence. Incorporating CRISPR-Cas machinery. To create bacterial biosensors that specifically detect mutant KRAS, the research team harnessed the bacteria's own CRISPR-Cas machinery. They directed the CRISPR-Cas system to selectively target and cleave wild-type KRAS, which would result in the death of any bacteria acquiring this gene. This approach effectively discriminated between normal and tumor DNA. In vivo testing and challenges, the researchers conducted in vivo experiments by administering the biosensor bacteria via enema to mice with and without engineered colorectal tumors. The results confirmed the bacteria's ability to signal the presence of engineered colorectal cancer in mice, demonstrating the potential for in vivo diagnosis. However, the translation of these biosensors to humans presents challenges. Human colorectal tumors do not naturally contain an antibiotic resistance gene, necessitating adjustments to the biosensor strategy. The researchers reconfigured the biosensors to detect natural tumor DNA with the KRAS mutation by incorporating a repressor gene inside the KRAS homology arms. This gene prevented the expression of a downstream KNR gene. As bacteria exchanged their KRAS DNA for the tumor's KRAS, the repressor was lost, allowing the antibiotic resistance gene to be expressed. The technique was named CATCH, standing for Cellular Assay for Targeted CRISPR-Discriminated Horizontal Gene Transfer. Towards clinical application. Despite the promising results, caution is advised. David Rigler, a microbiome researcher at Imperial College London, stressed that these systems are not yet ready for clinical use. The research team is actively working to enhance the biosensor's sensitivity to natural tumor DNA within the complex environment of the colon. Additionally, concerns about administering antibiotic-resistant bacteria to humans are being addressed by developing alternative methods for biosensors to signal the presence of mutant KRAS. The ultimate goal is to deliver these biosensor bacteria orally, allowing them to survive the journey through the digestive system and report their findings on the other side. Potential applications and future prospects. The potential applications of engineered biosensors extend beyond colorectal cancer detection. These biosensors could be designed to trigger the production of genotype-specific small molecule therapeutics, delivering treatment precisely where it is needed. They might also be tailored to detect and respond to various oncogenic mutations and challenging infections, such as Clostridium difficile. This prospect of combining diagnosis and therapy in a single system is a significant advantage of engineered biosensors. In conclusion, the development of bacterial biosensors for early colorectal cancer detection represents a significant step forward in the field of medical diagnostics. While there are challenges to overcome before clinical implementation, the potential to revolutionize cancer diagnosis and open new avenues for targeted therapy is an exciting prospect. The marriage of synthetic biology and medical research promises to make a meaningful impact on healthcare in the near future. Thanks for watching. Please see the citation in the description.